In progress, that sounds good. Yeah, sounds, so this is shared. Okay, sounds good. Thank you so much. Thank you. Yeah, okay, take care. Thank you. Okay, sure. Hello, everybody. Can you hear me in the back? Okay. So oh, should be okay. Thank you. Okay. Or is that working? Oh, that's working. Good. So this is the last class. Then next week is a study meeting. Little refreshing. What? Oh, okay. Okay, sorry about that. Oh, Zoom people, can you hear me? Yes. Okay, thank you. So anyway, let's get started. So this will be about how to modify genetically cells and organisms. So again, these objectives should make it a little bit more easy what to focus on. And so I don't want to read this, but like highlights on each slide and so. So first thing is why we want to study genetically modified cells or organisms. Why do we use that? Is we want to have a distinction between correlation and causative meanings of results. And so as an example here, I bring here from aging studies that a protein is increasing during age. So it's less abundant in young organism cells, and it will increase with age. So this means it's a correlation with age, but you don't know necessarily if this protein has anything to do with aging. So what you would need to do is getting to terminate, to delete this gene encoding this protein A. And so you create this knockout animals, cells, or whatever, and then you will repeat this lifespan studies, and you can see or expect, for example, that cells lacking this protein will have, for example, oh, thank you so much. Thank you. Thank you. Take care. So cells or organism will have a shorter lifespan or, for example, a longer lifespan. This means that the absence of this protein has a causative effect on the lifespan. If you don't see any change in lifespan, so it's most likely just a correlation. So this is one of the reasons why we want to create knockout cells or organisms. So there are a couple different chapters here. One would be we start off with genetic analysis of mutations. So when we study genetics or cloning and those things, we would have, for example, at the start off, we would have a mutant organism, mutant cells, mutant organisms. So this means what's written on the left top part, genes are not all, genes are missing, altered. So that means there's something missing in these cells, and that means it's a mutant. So in comparison to wild type means this mutant gene would be completely present and working as a functional gene. So we have these two mutant organisms. Then you can do a screening of, for example, DNA libraries. I will briefly mention this later to obtain the gene. So this means gene cloning. So once you have a piece of DNA fragment that can complement the mutant phenotype. This means, for example, mutant phenotype could mean it's like there's a stress response, it's sensitive to heat, for example. And once you clone the gene, the wild type gene, this heat sensitivity should be rescued. So these mutant cells should no longer be sensitive to heat. So this is how you would clone this gene. You would determine the DNA sequence to get the identity of this gene, because before you just have a piece of DNA fragment, you don't know what it is. So you do DNA sequencing. Once you get to know what this is, then you know, okay, it's similar to these and these genes, for example. So you know approximately what to look for, but it may also be completely unknown. So it's just, you know, it's a gene, okay, it's expressing a protein, but you don't know what it does. So one further work would be to look for localization. So to see, okay, this novel protein will be localized in, for example, nucleus, mitochondria, and so on. You may do biochemical studies. Say protein, this unknown protein may have similarity to, for example, a DNA helicase. So DNA helicases unwind DNA. So you do in a reaction tube a reaction where you look for unwinding of DNA pieces with purified protein of this unknown protein. So this would be going from a mutant organism to the cloned gene to determine what the protein does. You can also go the reverse orientation. So, for example, you may purify protein complexes. You identify the proteins in this complex, and then you would find, for example, novel proteins in this protein complex. Then you would obtain, for example, the gene, and you would create mutant organism to see, okay, this novel protein, has it any function for life, for other like stress response and so on. Okay, so two different direction here, going from mutant organism cell to cloned gene to the protein, study that, or coming from a, for example, novel protein isolated in a protein complex isolation scheme, coming from here to the cloned gene, and then you want to create mutants for example. Okay, so now in the next couple slides, I will focus on a couple words you use in genetics. One would be genotype, phenotype. So you see here on the right side these pictures of identical twins. This means once you have a fertilized egg, it will at some stage, I think that's before the blastocyte, it will divide in two parts, but this is not yet at the stage where it's differentiated. And then you would develop two organisms that are coming from the same fertilized egg. And so what you see here, at the first look, I mean, okay, identical twins, but they look a little bit different. So most likely they were exposed to different environments. They maybe were eating different food, different life situation. So that means appearance looks different. There could be metabolic, could be different. So that's a phenotype. Those two guys have the same DNA, or most likely say, when you look, when you don't talk about cancer or about, they have most likely 99.99% the same DNA. But when you look closely, and maybe also if you do blood analysis or something, you may see some different metabolic values. And so different phenotype means different gene expression, protein modifications, protein activity, and so epigenetics. That might be different in those two people. Okay, so genotype, phenotype. So you want to know what this genotype means, referring to the DNA, whereas genotype, phenotype means what's basically expressed from the DNA, and that could be very different under various situations. Again, here, examples for changes in the genotype would be alteration in its sequence. Parts could be missing, duplicated. Phenotype would be, for example, cells with mutations are growing slowly. Animals with a disease have a shorter lifespan, so that's a phenotype. Also, for example, susceptibility to stress factors, or you name it. So this would be phenotype. When we take a closer look on the DNA level and mutations, mutations means there is a change, could be a change in directly the sequence of the affected gene. So this could be on the left side, you see a gene, again, consists of an open reading frame. This is from start codon to the stop codons, the sequence, triple codons, that encodes the protein, but regulatory sequences are ahead of the open reading frames, the promoter. Sometimes they could also be placed downstream regulatory sequences in mammalian genes. So we would, for example, if a mutation is in the promoter area, there could be, for example, constitutively high expression, or it could be extremely low expressed, or it could be, for example, if it's developmentally regulated, it could be expressed at the wrong time, and so. So that would be the effect of mutations in the promoter. If there are mutations in the open reading frame, we would see, for example, point mutations. There could be, for example, some proteins like phosphorylated, for example, to get active. So there could be, for example, this phosphorylation site could be mutated, so it's not activated, for example. There could be portions deleted. So, for example, yeah, for example, ER retention signal KDEL, for example, if that is missing, it's no longer retained in the ER, for example. So those things. Rearrangement would be another way. These could have effect on, for example, expression level, enzyme activity, how proteins interact with each other, also with DNA, RNA, for example, or with the membrane, protein localization. So various examples what could be changed. And again, mutation in the promoter would have an effect on, for example, expression level at all, or also expression at times during development, for example, or something. And so these are effects directly related to the gene, but there would also be other RNAs, RNAs like microRNAs, they affect expression also of a gene. MicroRNAs interfere with the messenger RNA of this specific gene. So if those are, for example, not expressed, so there might be changes in expression level. Messenger RNA would be, for example, of this specific gene would be high or low, for example. Okay, so you just want to have an overall scheme of this to memorize that. Next one would be, so this was genotype, phenotype. Next, what words would be dominant and recessive mutations. So usually when we look in somatic cells, for example, everything comes in pairs. So there are, if you look at a gene, it comes in pairs because there are two chromosomes of, so like there are two chromosomes of, for example, chromosome 1, chromosome 2, and so every one is duplicated except X and Y, or yeah, for example, these chromosomes, they would be only once present. But so normally it would be, every gene would be duplicated because it's present on each duplicated chromosome. And so if there's no change in, for example, sequence or no mutation, we call that wild type. So that would hopefully be like most of our genes would be wild type. So it could happen that one copy of these is mutated. So when we look on the right side, so one copy is mutated and it says recessive. So this means the wild type gene can complement for the defect of the recessive gene. So there is a mutant gene that may also express this protein, but the wild type gene can complement for this defect. So it's expressing functional proteins, so, and that will compensate for the mutation in this other gene. sister gene. And so you wouldn't see any effect when you look at, say, at this so wild type and recessive mutation. When you look at these animal cells, you would not see any effect on the phenotype. So it would look like wild type. Only if both genes are affected by the same mutations, then you would see an effect on the phenotype. So it would look like a mutant. A different scenario with a dominant mutation or dominant mutated genes. So this means although there is a wild type gene, you would still see the mutant phenotype. So it doesn't matter, okay, there's wild type protein produced, but the mutant protein, for example, has overrules basically the wild type protein. That means dominant. And again, if both are mutated, of course, then you see the phenotype. But you want to memorize recessive mutation means if wild type gene and recessive mutated gene comes together as a pair, you would see the wild type phenotype. Unlike with the version where the dominant mutated gene comes together with the wild type, you would still see the mutation. So in this case, wild type protein does not compensate for the loss. So just memorize those two cases. These are two examples, dominant recessive mutations, disease, Huntington disease, for example. You can see here only, so if one gene has a mutation, one Huntington gene has a mutation, this individual is already affected by the disease. Unlike when you look at recessive mutation, you would see only an effect like the disease appears if both genes are mutated. So if just one gene is mutated, it's called carrier person. So it carries the mutation, but since this is recessive, you would not see the phenotype of the disease. So that's an example how you would see that with in human disease. Okay, so you want to memorize this dominant, recessive, genotype, phenotype. So when we look at, so for example, I think human genome has 20,000 genes. I do a little bit studies with yeast, so they have 6,000 genes. And so when you delete individually 4,000, I think 500 or so genes, you have 4,500 individual yeast strains, and delete those genes individually, you would see no effect on growth. So this means those would be non-essential genes. So when we look here on the left side, we see here A, B, C. So this could be like metabolic steps, for example. So when you go from A to B, you need the product of gene 1. And from B to C, you need product of gene 2. When you delete gene 1 and you will see cells are not viable, not living, then you know there's just one pathway, this ABC, and well, cells are not viable. So this means gene 1 is essential. If you knock out this gene, you will see cells are not viable, not living. In comparison, when you knock out here on the right side here, gene 1, and you see those cells are living, viable. So that means there has to be at least one parallel pathway. So you see A goes to X and then to C, for example. So since there is another pathway, that would result when you knock out gene 1, you see it's viable. And so you have now, I mean, you can extend this. There could be one other pathway, there could be 2, 3, whatever. So, okay, so essential means if you knock out the gene, cells are not living. That means there is just this one pathway. If you knock out a gene and cells are living, then you know there has to be at least one other pathway that's in parallel to the, for example, ABC pathway. Okay. Okay. So these were examples of recessive mutations. Mostly when you study, for example, you knock out the gene, nothing happens, for example. I mean, there could be an effect somehow, a mild effect of something, stress response. But if a knockout results in a lethal phenotype, so you would not be able to study this gene. So what you need to do is creating a condition that makes a viable and a non-viable phenotype. And that's what you see here on the left side, you would here the scheme with the sequence here, you would use a mutagene to change, to alter the DNA sequence. And you would use this mutagene. This could be a chemical, for example. This could be, for example, UV light, could be radiation, could be. So you would use this as at a very low level to introduce a low level of mutations. So you would introduce just a single mutation here to change just the sequence of one nucleotide. So you see here, there would be one pair, thymidine, adenine. So there would be, say, adenine would be changed to thymidine, and then following here after replication, you see that the sequence has changed now. So when you do this, so codon usage would be, for example, altered in this sequence. This would be a way to create mutants that, in this case here, would grow at room temperature, but if you increase temperature, they would not grow. So you have the same DNA, but under different conditions, you have a viable condition and a non-viable condition. So again, at normal growth would be at room temperature, 20, or whatever, 23 degrees Celsius. If you increase the temperature, for example, here 30, 36 or some high temperature, you would see cells are not growing anymore. So so you can, maybe I think I have, yeah. When you, for example, in this example, we are using yeast, but you can also use animals. You can use, for example, C. elegans, worms, for example, is often studied, uh, or flies, for example. So in this case, you use yeast cells, and you would add a low amount of this mutagen, like a chemical that would interfere with the DNA. It would change the DNA sequence. But you would add just a low level of this mutagen. If you use, I'm sorry, if you use a lot of this mutagen, you would introduce a lot of mutations. So that wouldn't help you to create this conditional mutations. So when you plate out yeast cells treated with a mutagen on plates, you would see a certain number of colonies at room temperature. If you transfer those colonies to two plates now, it's called replica plating, and so, but you have the same colonies at the same place on the plate. You would see at room temperature, there would be one colony would grow, but at high temperatures, the same colony would not grow here. You can see, I don't know this pointer. You see, so one colony would not result in growth. So in this example, we have a temperature-sensitive mutation or mutant that grows at room temperature, but not at high temperature. Maybe this would be a little bit more in detail. Temperature-sensitive mutants means that you introduce, for example, a point mutation in a single gene that affects cell growth. So it could, at normal temperature, at room temperature, this protein would fold properly. There would not be anything to see. It would look like wild type protein, properly folded, proper function. So no phenotype that you would see. Looks like wild type. If you increase the temperature, so we talk about the same cells, this protein with the point mutation will unfold. So if it's unfolded, hydrophobic sequences, for example, would be exposed to the outer side, and this would trigger degradation of this protein. So we use here, again, the same strain, yeast strain, same cells, cultivating, growing them at room temperature. You would see no phenotype. They grow normal because protein is properly folded and is functioning. Whereas at high temperature, this same protein gets unfolded and mutant has now a phenotype, for example, stops growing. So, okay, so using this strain we can now, or this strategy, we can now create temperature sensitive mutants. So if we come back to this scheme where we plate out cells on agar plates and then looking for colonies that will appear at normal temperature, but not at high temperature, you would not just see one colony. You probably will see multiple colonies behaving in this way. Once you isolated this individual colonies, so let's say you have 50 different colonies, but you don't know is in these 50 different colonies just one gene affected, so with this point notation, or are there multiple genes having the same effect? So what you do is, it's called complementation analysis. And so it's maybe looks a little bit complex, but what you do is you create diploid strains. So maybe I have to add information here that for the mutagenesis in the previous slide here, when you add the mutagene, you use haploid yeast strains. So that means yeast has a certain number of chromosomes, and in the haploid strain, there's just one set of chromosomes. Whereas when we look at diploid strains, you have two sets of each chromosome is duplicated. So what you do in the complementation analysis is you create from haploid yeast strains, you create diploid. In doing so, you need, I mean, this shouldn't confuse you. You have one set of mutant cells, for example. It's called mating type. So they would be all, so in yeast it's not male, female. In yeast would be A and alpha mating type. So they would all be A. To create the diploid strains, you need to have A and alpha. Then you can cross those and then you create diploid cells. So this shouldn't confuse you. I mean, you can make, when you have a set of of mutant strains with a mating type A, you can convert this to alpha, for example. I don't want to go into detail here, but so you just convert it A and to alpha. Then you have two sets of mutant strains. One is mating type A and one is mating type alpha. So identical strains or pairs of strains, but mating type A or alpha. And so what you do is you would cross now all the mutant strains, you would cross those with each other. So this is a scheme. You have mutant 1, 2, 3, 4, 5, and you have here mating type A would be in the first row, in the column here, mating type alpha, and there are, in this case, 25 different combinations of what you can create, how many different crosses you have. So when you do this, you have 25 different mutant strains now. And this is what you see here on the right side. You would cross here mutant strain 1 with 2, for example. You would cross those. You would create diploid strains. So that's the scheme what I showed you before. And you would plate out these on agar plates. So at room temperature, again, they should all grow. But the difference is now when you plate those cells at high temperature, there could be situation where these colonies would still be growing, or there would be no growth. Not any colony would grow. So what does this mean? When you look on the left side here in this scheme, you see that the mutations of these two genes are in different genes. So here we have X and Y. This is not in the same gene is affected, but these are different genes. So if you create diploid strains, you would have for example, mutant gene. So for example, the Y gene would be mutated in one copy, but the other copy would be wild type. Similar here, you have the X mutation here. That's the yellow bar, but the corresponding sister gene would be wild type. So in each case, the wild type gene would complement the recessive mutation in the other, the mutation in the other gene. So this means X and Y mutant would be located, the mutations would be located in different genes. A different situation is on the right side, where you do the same, cross the strains, get diploid colony, a diploid strain at room temperature. But when you plate those cells at high temperature, none would grow. So this means X and Z, so these two mutations, what you see here at the bottom, they would be in the same gene. So because if both genes are mutated and they are in the same gene, they cannot complement each other because both genes are affected by mutations. So this is the way how you would determine how many genes would be affected by your mutant screen. What I said before, when you do the mutagenesis, you may get, for example, 50 different colonies, but there may only be 2 genes affected in these 50 colonies. So this is the way how you determine this. And this is again, we deal here with recessive mutations, not with dominant. That wouldn't work. Okay, so another thing what we can determine is the order of events, like steps in biosynthetic pathways. For example, you have a mutation in gene A or a mutation in gene B, for example, depending on what metabolic intermediate will increase. You can say, okay, A comes before B or the other way. So I just want to highlight, depending on what metabolic intermediate will increase, then you can say, okay, it's in this order, or it could be 1, 3, 2, for example. Okay, so that's this slide is very important. So when you so you can bring together different mutations, and they may have different effects on cell growth. So one could be like one mutation is suppressing another mutation, or if two mutations come together, they will enhance the effect. So in the first case, suppression means you have, in the wild type scenario, you have A and B, so it's a perfect, for example, key lock principle, everything fits together. If you change either key or lock, so you would create either one or the other mutant that would make cells, for example, growing a little bit more slowly. But if you bring together both mutants, so on the right side you have mutant A and B, you bring those together, those will be growing or behaving better than the single mutants. You may not reach the level of what's wild type, but suppression means the combination of the two mutants in one organism or cell may result in a better condition compared to the single mutants, but they may not reach wild type level. In contrast, what we see here, it's B and C here down, synthetic lethality. So we have the same situation. We have two mutants. We combine. First, we have two mutants. The single mutants, they would have, for example, mild effect on, for example, cell growth. But if you combine the two mutants in one organism, one cell, you would see a severe effect on cell growth. They may not grow at all or very slowly. So this means it's called synthetic lethality. And so this also means that there would not be, for example, if we talk here again about two different pathways, for example, so we have here, for example, only two pathways. There would not be, for example, a third pathway when we talk what I talked before about essential, non-essential genes. So again, synthetic lethality in this case here is single mutants would have a mild effect on, for example, cell growth. But if you put together these two mutations in one organism, you would see a very severe effect. They would not grow at all or very slowly. Similar like what we see in the bottom row, if for example gene A is needed to make for example a certain product, also gene B, if you delete either A or B or its absent, you would not see a major effect on, for example, cell growth, because the product would always be produced. But if you knock out both genes, A and B, in the same cell, you would see the end product is missing. And so so this is another example of synthetic lethality, where you where you do not synthesize, for example, a common end product. So again, suppression, single mutants have a, for example, some mild effect. if you combine these single mutants, they would grow better. They suppress each other's mutation phenotype, but they may not grow as well as wild type. So this is suppression. Synthetic lethality means you have two mutations, and combining those two will result in a severe phenotype. Cells would, for example, not grow at all or very slowly. Okay, so memorize this. Next part would be DNA cloning and how to characterize mutations. Cloning means that you want to amplify a certain DNA fragment. This could be a gene or some piece of DNA, and you have to amplify this piece of DNA. So you have to, it could also be you have only small parts of this DNA, and you have to get a reasonable amount of DNA so you can make, for example, mutagenesis or something else. So what you do is you put this piece of DNA fragment in this circular structure called vector or plasmid. So you would insert this in this plasmid and create recombinant DNA because it has now the DNA fragment and the plasmid. So it contains two different parts. This plasmid DNA allows basically to amplify the DNA fragment to a certain amount. So you would introduce the recombinant DNA into host cells. This could be bacteria. This could also be insect cells, yeast, for example. So you would create a high number of plasmid DNA, and then you would isolate this plasmid DNA sequence. You can manipulate, mutagenize. Okay, so we go now. First part is we have plasmid DNA and we have the DNA fragment. Or we don't even have the DNA fragment, for example. So in order to create DNA fragments or to open the plasmid DNA, we use restriction enzymes. Restriction enzymes were originally found in bacteria, and so they cut DNA at a certain sequence. What you see here is, for example, GAATTC. So this is called a palindromic sequence. You want to memorize what the palindromic sequence is. Because when you look at the bottom strand, you would see reverse. When you read in the reverse orientation, you will see GAATTC. So when you look first strand and the second strand, and you read that in reverse orientation, you will see it's the same sequence. So this means palindromic sequence. This is one example of a restriction enzyme, ECOR1 or Escherichia coli R1. So I think that was one of the first enzymes that was identified, and it was actually identified in bacterial cells. Escherichia coli is a bacterium, and so produces this enzyme. The function of this restriction enzyme in bacteria is to digest foreign DNA. So if foreign DNA comes into bacteria, for example, bacteriophages, these foreign DNA will be digested. In order to prevent that this enzyme will digest the DNA from the bacterium itself, bacterial DNA is methylated. You remember maybe gene expression, so you can have methylated DNA, and in this case it's also methylated methyl groups on the bacterial chromosome. So this ECOA1 enzyme will not digest its own DNA, only foreign DNA. So again, if you palindromic sequence, when you read bottom and top strands, it's in reverse orientation, it's the same sequence. When you use this enzyme, you will create ends with, it's called sticky ends. So you have single-stranded DNA as overhang, TTAA, for example, here, AATT. These are complementary to each other. So, yeah, and this was, you can see here, I mean, this goes back to, I don't know, 60, 70. And I think the insulin gene was one of the first genes that was cloned. Okay, so memorize what's palindromic. So what does this mean? There's not just one restriction enzyme. You can go to like a catalog, and not Amazon, but maybe no, a catalog, and you can buy hundreds of or lots of different enzymes. This is just a list of different enzymes, what they would recognize. Don't need to study this for exam, but you see here different sequences, different lengths. There's, for example, not one here. This sequence exists very rarely in chromosomal sequence. So, for example, with the yeast chromosome, if you use this, you would create, for example, 2, 3 fragments of a huge chromosome. So it's not very often present. Others would be present every other 10 base pairs or so. I mean, very often, and BAMH1, for example. Okay, so yeah, so different enzymes with different endings, 5' sticky end, or for example, blunt end means there's no overhang, for example. So blunt end, for example, is not good for cloning. A sticky end with an overhang would be much better for cloning purposes. So that's exactly the slide, what I mean here. When you cut DNA, in this case ECOA1, you have this sticky overhangs, TTAA, for example, and then you want to bring together DNA fragment with the plasmid DNA, vector DNA. So these sequences TTAA and here AATT are complementary to each other, so they will anneal, you see here, they will find each other, and then you will see here base pairs. But there will be a little gap is left here because there would be the phosphate group of the last of the 5' end, and this would not be connected to the plasmid DNA. So this gap is not good for when you bring this into bacterial cells. You may induce rearrangement of the plasmid. So what you do is you will seal this gap by using a ligase. It's called, in this case, T4 DNA ligase. This comes from a virus, but the purpose is to seal this little gap here. So it's connected and it's double-stranded throughout the sequence. So there's no gap. On the right side, you can see an example of a plasmid. There are three different parts that this plasmid contains. So there would be a replication origin is needed to amplify the DNA. If you bring in just 1-2 copies of the plasmid DNA into the host cell, it has to be amplified. So it needs to be, DNA needs to be duplicated. And this duplication process is starting at some point in the DNA, and that's the replication origin. So that's necessary. The same is with the marker gene to select cells. If you do, it's called transformation. If you bring DNA into cells, this will only happen to maybe a few cells out of millions. If you plate cells on a plate, you would not know which cells have the DNA or not. So if you use this marker gene, in this case, it's like a antibiotic resistance gene, ampicillin. So if you use that, you can select cells containing the plasmid DNA. So number 3 would be many different plasmid DNAs have, for convenience, they have a cloning site, a multi-cloning site. You don't only have eCOR1, you have multiple different sites. It makes life easier when you do cloning. You can do different cuts. So it's just for convenience. So you want to memorize replication origin and the marker gene are necessary. The multi-cloning site is just for convenience, not necessary. So when I say it's important, you know what I mean. Okay, so this is when you have, in the previous slide, when you have a certain amount of DNA present, and then you do restriction enzyme, you do this ligation, and so on. But sometimes you may have very tiny amounts of DNA. This could be, for example, you could have DNA coming out from a single cell. You can have, for example, looking at single like stem cells or whatever. Techniques are here to look at single cells and so on. To amplify, in this case, DNA, you use the technique called polymerase chain reaction. You may have heard, of course, this during COVID, for example, you go for the testing. So this involves the PCR reaction. That was not only PCR, that was also using a reverse transcriptase to go from RNA to a DNA and then amplifying the virus DNA. So what you do here during a polymerase chain reaction is to go from 1 molecule to 2 to 4 to 8, 16, and so on. So you amplify it by steps 2, ideally, but it's probably lower. One caveat or one problem is the DNA strand is very difficult to get separated. what you need is single-strand DNA, because what you need here is to synthesize the complementary strand with an enzyme. If you have a duplex strand, it will not help you. So what you do here, what you need is high temperature. You see here 94 degrees. This is basically boiling water. There wouldn't be any like living, like if you have regular cells, 94 degrees, everything would be dead, denatured, there wouldn't be anything working, so you can't use. So there was somebody, who was it? No, this was Tom Brook here. This goes, you can see here, 1965, studied spring, like how to say, like a hot springs, hot boiling water. And he could see, oh, there are some living cells in this hot spring. So how do they survive? And so one thing he isolated was a enzyme that duplicates DNA. And this is Tec polymerase for Thermo aquaticus. If I say Thermus aquaticus, that's a bacterium. And so this is what's known today as Tec polymerase. So this polymerase is able to synthesize DNA in boiling water. Other living material would not be able to survive those conditions. So this was 1965. But Carey Mullis, so I guess this was when they also for cloning of a gene, and so thought, okay, what can we use to synthesize DNA or to overcome this barrier with, so we have this hot step with boiling water, and then what would we add here? And then thought, okay, we use this enzyme polymerase from this bacterium, and that would survive in these cycles. Here, what you can see here, when you need boiling temperature to get the strands separated, so to get single-stranded DNA. Maybe so. Again, the first step here, double-stranded DNA. You have high temperature, like 94 degrees. You get single-stranded DNA. Now you need to synthesize DNA. You need a little piece of DNA that will anneal, that will prime to the single-strand DNA. So polymerases in general, they need like these little primers. It's called primers or oligos as a start point. There would only be this is called terminal transferase. That's enzyme that will add nucleotides to an open end. Doesn't need any primer. But usually polymerases, DNA polymerases in general, they would need these little pieces of DNA. It's called primer, and that's where DNA synthesis would start. So you see here like this blue, dark blue, light blue boxes, and these are the primers. And so when you add now the enzyme, it will extend, it will create the parent strand, and you will create a duplex strand. So when you anneal these primers, you would have to do this at a certain temperature. And when you design these little primers, they could be, for example, 15, 20 nucleotides or so. You would design this using a program that shows you, for example, melting temperature. So you want to have a melting temperature that, so you would have, for example, 50% annealed and 50% loose, not annealed. So these conditions, for example, the program would tell you the temperature, what you have to select to properly anneal the primers. This is standard procedures when you go, for example, IDT is a company that produces primers, oligos, and so on. And this is a standard procedure to order these primers. And you have to select the location of these primers. So they should be complementary to a certain sequence and so on. And another important part is with the PCR reaction, you can only amplify a certain length of DNA. So if you, for example, 1 kilobase, 2 kilobases, 3 kilobase, there are specialized polymerases. You can amplify up to 10, 20 kilobase, but these are specialized polymerases for PCR. And conventional, you would use maybe amplify a couple kilobase or something like this. Okay, so to repeat this, you here have a temperature where you separate the strands, creating single-strand DNA. Then you anneal, it's called annealing step. The primers, the little oligos, would look for homology on the single-strand DNA. And you use 2 primers, you can see. And then you would have the amplification, say synthesis step at higher temperature, usually 72, 68 degrees Celsius. And you have a first step, you have a second step. And after the second step, you can see you form a discrete DNA fragment. And this will be amplified when you go, usually you go like 30 cycles of this, denaturing, annealing, synthesis. So you go 30 times this, and then you will see a certain amount of end material. Okay, so this is, you just want to memorize this, what's say denaturing, annealing, synthesis, so what's needed. And to facilitate cloning into a plasmid, so you could have, for example, overhangs within restriction site, for example, and this would facilitate, say, when you look downstream, you have a restriction site here at the end that would make it easier for the cloning, for example. Okay, so just memorize what denaturing, annealing, synthesis, the DNA polymerase, what kind of feature does it need, and so. Okay, so this is PCR. Once we have the DNA fragment ligated into the plasmid, we have to amplify this in a host cell. And I told you there are two important parts in the plasmid. One is the origin of replication. To amplify the DNA, it has to be synthesized, DNA replication. And the first part would be to select those cells that are having the plasmid. So there will be only a few cells obtaining the plasmid, but the remaining million of cells will not have the plasmid. If you plate cells on a plate, you will just see like a lawn of cells, but don't know which one has the plasmid. So these cells, the plasmid has a selection marker, in this case, an antibiotic resistance gene. So you would plate cells on a medium containing an antibiotic. So in this case, it's ampicillin. And what's the restriction, the ampicillin resistance gene, what it does is it will digest the antibiotic. The antibiotics, the ampicillin, would interfere with the cell wall synthesis. So usually bacterial cells, they would just keep growing and dividing, but the antibiotic would interfere with the cell wall, so it would make the bacterial cells to lyse, to explode, basically. Unless the cell contains the plasmid with the antibiotic resistance gene. So this resistance gene produces a protein that will digest the antibiotics. So those cells can grow, and those cells will form a colony. So this is how you select cells, those cells containing the plasmid. Okay. Sorry about. So once you have the DNA, you isolate, so you obtain a colony on the plates and you restreak those cells to get single colonies, and you would put these like a single colonies and in a tube with medium, also containing ampicillin. So you would grow up a certain number of cells and you would isolate the DNA. This is also a standard procedure. We would use actually sodium hydroxide to open the cells. There are certain, it's called kits where you can, for isolation of plasmid DNA and so. Once you have the DNA, you want to look at size of the, for example, DNA fragment that you have cloned. So you would, for example, digest the DNA with a restriction enzyme, and then you would run it out on an agarose gel. So these usually we are talking here about DNA fragments of maybe 1, 2, 4, 5 kilobase or something. So you would use a, for example, 1% agarose gel, and when you apply an electric field to this loaded DNA, it will move through the gel with different speeds. So short pieces of DNA will migrate fast compared to large pieces of DNA. They will not go well through the agarose. Agarose contains like pores where DNA goes in and out. And so if the DNA would be, for example, large, I mean, it would be more difficult to get through. Electric field is because DNA would be negatively charged and would migrate to the positive electrode. Here, again, you see DNA contains on the surface phosphate groups, which are negatively charged. And so this makes it moving to the positive electrode. And once you finished running out the DNA, you want to look at the DNA. And this is by using a chemical that will insert into the DNA. It's called intercalate into DNA. And this is, in this case, for example, ethidium bromide, which will insert into between the base pairs. And so under UV light, you will see bands, lines, bands appearing in the gel, and that would indicate size of DNA fragments. You would run out on the left side here, it's called DNA ladder. So it's a size marker, and so you know how big, how long would be your isolated DNA fragment. Okay, again, you want to know DNA is negatively charged and will go to the positive electrode. So this was just overall DNA cloning. And when we go back to the identification of a mutant gene, or what is the identity of the mutant gene, we have to isolate the functional gene. So we would use this mutant that can grow at room temperature, but does not grow, for example, at high temperature. So this temperature sensitive mutant. And we have identified, for example, okay, there's one location in the genome that has a mutation. And so we don't know yet what's the identity of this gene. So we would introduce a functional gene that can complement the defect. So in this case, these cells with the plasmid DNA, with the functional gene C, for example, here, because there's a mutation in gene C on the chromosome. So it will grow at room temperature, but also at high temperature, because now in this case here at the bottom, we have the functional gene in the cell, so it can complement the defect, the temperature sensitive defect. So there are, I mean, this is yeah, to know. I mean, there are DNA libraries, sorry, my libraries collection of genes. They would come, there are two versions. One would be genomic DNA library, which is a collection of DNA fragments throughout the whole genome. So that would cover all genes present in the DNA. In contrast, complementary DNA library or cDNA library is a reflection of the RNA present in the cell. So that could be different. When we look here, we have here comparing tissue A versus tissue B. There are different genes expressed. So we would obtain different cDNA library because we will use as a starting material messenger RNA. In contrast, genomic DNA would be the same. So there would be the same genomic DNA library compared to the cDNA library would be different because there are different genes expressed, tissue A versus tissue B. Okay, so you want to know what's genomic DNA library versus cDNA. So genomic would be same DNA versus cDNA. That depends on what messenger RNA, what kind of conditions tissue was used. This is a little bit complex, but this is how you would create a cDNA collection library, complementary. So you would start off with messenger RNA. In general, RNA is not very stable. You just need to sneeze and it will degrade. So it's you cannot really work with this. So what you do is you convert the RNA to a DNA. And so you would use here on the right side in the box, you can see RNA is converted to an RNA DNA hybrid using reverse transcriptase. So the reverse transcriptase enzyme will synthesize the DNA strand using the RNA as a template. This is also shown here on the left side. You use a oligo DT as a primer. This is this green bar here. And then you synthesize the corresponding, the complementary DNA sequence using RNA as a template. Also, this version, RNA DNA, is not very stable. Also, you would replace the RNA strand with the DNA strand. So in this case, RNA is degraded, but then you have the synthesis of the complementary DNA strand, and then you have a duplex DNA. So both strands are DNA, and this you can use for cloning purposes to put it in a plasmid. So you want to memorize reverse transcriptase produces a DNA RNA hybrid using an RNA template. Okay. Very often viruses contain encode also reverse transcriptase because they contain an RNA, and to convert this to DNA that can then integrate into the genome. So this is where it comes from. So reverse transcriptase is used to convert RNA into DNA, basically. Okay, this is cDNA library. In contrast, genomic DNA library is a collection of different DNA fragments. So this could be thousands and thousands DNA fragments. They would overlap. Here, what you see here on the right side, you have genomic DNA that you digest with restriction enzymes, but you create it in a way that this fragment would overlap. So it's not completely digested. It's one fragment would show up sequence in another fragment. So it's that's called overlapping. And you would put these digested DNA fragments of a genome, you would put this into plasmid DNA. This is also yeast specific and so, but what you see here, you have hundreds of plasmid DNA molecules and they may contain different DNA fragments from the genome. So it's a collection of DNA fragments. This library contains a representation of a whole genome within this plasmid DNA. So these small fragments together would represent an entire genome. You would put these plasmid DNA in a first step into bacteria and then into yeast. This is just for like a routine step to amplify the DNA first, and then you can use it in yeast. I mean, this is yeast as an example. You can also do similar things in, for example, other, like in worms, for example. Okay, so genomic DNA library is a collection of DNA fragments representing an entire genome. So, coming back to the temperature sensitive mutant, you would transform, it's called transformation, you would introduce this collection of plasmids containing different DNA fragments. You would introduce this into these yeast cells that can grow at room temperature, but not at high temperature. So here you have on the left side, you have the temperature sensitive yeast mutant. And on the right side, you have the collection of this plasmid DNA. You do a transformation, you introduce the DNA into the yeast cell, and you plate out these cells on an agar plate. In this case, you have a different way to select this plasmid DNA in the yeast cells. It's by lacking, in this case, uracil here, but this should not bother us. You select plasmid containing yeast cells. You obtain colonies on a plate at room temperature, okay? And then you would plate, or it's called replica plating. You use a little piece of tissue, like a tissue, and you would do you would press the first plate on this tissue, and then you take a fresh plate and hold it, press it on this tissue, on this velvet. So this is called replica plating. And then when you grow these new plates at high temperature, you may get colonies that will grow. The majority of the colonies may not grow. Why is this? So here we have three different examples. So we have a cell with gene A, B, C on the chromosome. Gene A has a mutation. B is not affected, C is not affected. So a genomic DNA library is a collection of different plasmids containing different DNA fragments. So in the left cell, you see here gene A is in the plasmid. But in this case, it's a functional gene A. And when you look here, these cells here with the gene A, they can grow at high temperature. When we look here, the two other cells, there's a gene B and a gene C in the plasmid that will not complement the defect in A. So they will not grow at high temperature. So this is a principle of complementation. So you have in the plasmid, gene A is a functional gene that will complement the defect in gene A on the chromosome, which has a mutation. If you have gene B or C in the plasmid, no effect on the complementation. So they will not grow at high temperature. So, I mean, this is called complementation of a defect when you introduce the functional gene that can complement a mutant gene. Okay, so this is how you select now. Now you have this gene A identified. Next step would be you want, all what you know is, okay, you have the DNA fragment, but you don't know what is it. Here comes in DNA sequencing. You determine the order of the nucleotides, and you do that by sequencing, which means you have 4 nucleotides here, A, G, C, T, and you would add some nucleotide that will stop synthesis. So here you would add a version of, for example, in here you would add a dideoxy-GTP to this reaction with the deoxy-GTP in low concentration. So it would stop at certain points along in different DNA fragments. And so you see here, for example, in the sequencing reaction, you synthesize the complementary strand of the DNA that needs to be sequenced, and it will stop here. For example, this is the first C here, or there would be another C. So say this is on the next slide. The nucleotide that will stop the synthesis is lacking the hydroxyl group in the ring structure, in the sugar. So that causes the synthesis to terminate. And when you do that, in a way that you add this modified nucleotide in a low level, so it will stop synthesis in various DNA fragments at various sites. So what you obtain here on the left side here, you can see here you will obtain a ladder like of different fragments of bands. When you start, I have to add here, when you start synthesis, you have a radioactive labeled DNA primer at the beginning. You see here at the top, you have a little piece of DNA, single-strand DNA that will anneal to a certain location, and this primer is radioactive labeled. So when you expose the DNA to an X-ray autoradiogram, it will appear like dark bands representing where the DNA fragments are. But again, since you add different concentration of this modified nucleotide, dideoxy ATP, for example, it will stop synthesis at various points. So therefore, you see multiple bands within the lane. If you add too much of this modified nucleotide, it will stop it will start synthesis of the complementary strands, but then it will stop. If you add too little, it will just synthesize, and it will you will not see this kind of ladder. So when you do that, certain concentration of this modified nucleotide, you do that with all four different A, G, T, C. You have four different lanes, and then it will you can read the sequence like here on the left side when you go top to bottom, for example, C, G, T, probably another T, A, T, and so on. You go in this way, so you can read out the sequence. This is also, I don't know if you study already for the exam. I said when we looked at DNA binding proteins, there was a technique called DNA footprinting, where you identify the exact location of a protein sitting on DNA. And that's exactly what you would run out in parallel to this footprinting. You would run out these four sequencing reactions. So you know exactly which nucleotide position the protein would bind. So just as a side information. So, I mean, this is a principle that was used, well, I mean, it's a standard principle also now, but I mean, this reaction type, what you use here, we have used this maybe 1995 or something, or I was using this thing. But what you today, when you do, you isolate the DNA, you look, okay, what primers do I need? Or are these conventional primers that already sites are present in the plasmid? And so you would drop off the DNA like in a like the boxes outside when you wait for the bus, for example, on the floor. You drop off your DNA samples at night. Then sequencing labs are picking up your samples, and they would use fluorescently labeled nucleotides, and it's in one reaction you would detect all four nucleotides, and this is how it would look like. So the next morning you would already have the DNA sequence. So I mean, this is standard, and so this was sequencing one DNA fragment. So you can do this about maybe 500 nucleotides, 600. This would already be very long. There are now methods available already. I don't know, maybe this was maybe 10 years or so. It's called whole genome sequencing. And you can use either just the DNA, or you can also use RNA produced from the cells and make DNA and then sequence that. So again, what we have genomic DNA library, cDNA, it goes the same way. Yeah, just for this is more or less information purpose. I mean that you know this exists whole genome sequencing, with using either DNA fragments or RNA. This is just how technically how it would look like. DNA fragments will be immobilized on a platform, and then you would also use fluorescently labeled nucleotides and go basically one by one reaction. So just that you know this exists, and yeah. Okay, knockout and transgenic. Knockout is if you if you delete a gene. Transgenic is often called when you want to overexpress something in animals. I go from the low level organism, that's yeast, where for example I said all, for example, 4,500 open reading frames were individually deleted. So there are 4,500 different yeast strains. And this was done by a PCR method. And in general, when you knock out a gene, you will replace the gene with a marker gene. So it's the same principle what I said before with the plasmid, with the antibiotic resistance marker, and you would select those cells containing the plasmid. In this case, you would select those cells with the knockout because the gene of interest is replaced with the marker gene. Doing this, you have to create like a PCR reaction using primers. Here you can see here primer 1 and 2 does not only contain sequence to amplify the marker gene, CAN-MX. This is kanamycin resistant gene. So this kanamycin resistant gene is here, this orange box here. This is on a plasmid, and you would design primers that partially will have homology looking to anneal with the kanamycin resistant gene, but they would also have overhangs. This is this purple color overhangs that target these marker gene to the gene of interest. So this is shown at the top here, target yeast gene. You can see here in purple and in green, this is the site where the primer sequence has homology, and the goal is to replace the blue colored intervening sequence with a marker gene. So you have here in the PCR reaction, the first part will be annealing. The primer will look homology with the kanamycin resistant gene, but then already in the second reaction, PCR reaction, you will see here there are overhangs with the homology to the target yeast gene. So the purple and the green will interact with the purple and green boxes here on the chromosome, and the target yeast gene, the blue box, will be replaced by the kanamycin resistant gene. This is shown on the next slide. When you do this knockout, you would do this in diploid cells. You don't want to do it in haploid, because if you do a knockout and you don't get any cells, you don't know, okay, it's because the gene was essential or not. So it will not give you the information. You do it in diploid cells, and there will be one copy will be knocked out. So again, this means this PCR product, the flanking sites will look for homology of the flanking areas of the gene of interest that needs to be knocked out. So you replace the gene of interest with the marker genes. You see here in the third cell from the top, you see here instead of the blue box, you can see the marker gene, the orange colored box is present in, so one copy is replaced. And this is just for the yeast. I mean, you can go diploid cells, you can produce haploid cells, then you can see, okay, do the haploid cells grow or not grow when you look at the gene knockouts, because now you have just one copy. And if this is, for example, not growing, then you know, okay, the gene knockout is affecting an essential gene. If it's not essential, you make a knockout, they will grow. Okay, so this is a principle that's also used for mammalian cells for the gene knockout. But when we look here, okay, mammalian cells, it's a little bit more complex, because mammalian cells do not have this efficient, or it's called recombination, looking for homology. If you have, say, DNA fragment that's used for, say, knockout of a certain gene, very often this DNA fragment would go to a different location in the mammalian genome. To distinguish between the location where it should be and other locations, you have two marker genes. So one is here, it's called neomycin resistant gene. That will, it's the same principle like what I said before, kanamycin resistant, replacing the gene of interest. Here, the orange colored boxes are the sequences that are flanking the neomycin resistant gene. And you see here on the left side, that will replace the gene of interest, or part of the gene of interest. In the DNA fragment that will be transformed, there is also another marker gene that will help to identify those cells where the gene of interest is modified, replaced, or deleted, and not the construct is going to a different place. So it's called thymidine kinase. And this thymidine kinase converts a drug, ganciclovir, to a toxic nucleotide. So if the thymidine kinase is present in the cell, it creates this toxic nucleotide. If there's a correct integration at the site of interest, the thymidine kinase is not present. So that's on the left side. If the construct will go some other place, the thymidine kinase will still be there. And so that makes this conversion to say, if the ganciclovir is present, it will cause the conversion to this toxic nucleotide. So at the bottom, what we see here, if it's at the correct place, cells are resistant to, it's called neomycin or G418, and ganciclovir. So because the toxic nucleotide is not formed, because the thymidine kinase is not present. In contrast, if the DNA fragment is somewhere else in the genome, thymidine kinase is present, and those are sensitive to ganciclovir. So these two different compounds so allow a dual step screening. The first one is with the G418 drug, just to see, okay, we get some transformants, but then you would screen those for the sensitivity to this ganciclovir, and if it's sensitive, then you know thymidine kinase is present, so it's most likely some other place. If it's resistant, then you know likely it will be at the right place, the knockout construct. Okay. Once you create knockout is most likely done in embryonic stem cells, and now it comes to creation of the knockout mouse, for example, in this case. So you would inject the embryonic stem cells. You would inject those into blastocytes. I guess you heard from the stem cell lecture from Dr. Freudenreich. So it's after 4 or 5 days, you can see this blastocyte. It's not yet differentiated. So you would inject these embryonic stem cells. You would implant this into a mouse, female mouse, and then you would do numerous crosses. The goal is to obtain a double knockout mouse so that both copies are present as knockouts. So this, of course, would so it means that from this method, you would also, the knockout would not be life-threatening. Let me say it in this way, that it could be, for example, a knockout could be, or a gene could be required during development. So you would not get a full-grown mouse, for example, if you have a double knockout. You may see this already in the heterozygous, meaning you have one gene knockout, but the parent, the sister, on the sister chromosome, you have the functional gene. You may already see some effect. Okay, so overall, what you should memorize is with, you don't need to memorize these steps here with the crossing, but what you should memorize is with this method, you create a mouse with the knockout of a specific gene in all tissues. So any tissue will be affected by this knockout, because you go from the blastocyte, basically you go from the beginning, you go on, start off with this knockout. Okay, so all tissues will have the knockout. In contrast, there are methods where you can create knockouts in specific tissues. This is with, for example, Cree lock system. So in this system, a gene knockout will be created only in specific tissues, or I mean, you can also use this method to do the knockout in the mouse at a later time point in development. So for example, the gene is required during early development, then you induce the knockout later, at a later stage, for example. So what you do here with the Cree lock system is on the left side, you see the gene most likely consists of different exons. So 1, 2, 3, a mammalian gene has areas that are showing up in the mRNA, but a lot of portions of this DNA, when it comes to the RNA, will be spliced out. So introns will be spliced out, and you will create a mature messenger RNA not having any introns. Okay, so you would, for example, design now a mouse with these LOXP sites flanking, for example, exon 2 here. And you do this same principle what I said before with the transformation, marker gene, and so on. Okay, so you would create a mouse having of the gene of interest would have flanking sites, LOXP sites, flanking sites of exon 2. So in different tissues, what you have the same genome, and you would have all in these different tissues exon 2 of your favorite gene will be flanked with the LOXP sites. Now you cross this mouse with a mouse expressing or having the Cree recombinase in the genome. This Cree recombinase recognizes these LOXP sites and splice out the intervening sequence. So the exon 2 will be spliced out. The Cree recombinase is recognizing the LOXP sites, and the exon 2 will be removed. So after the action of Cree recombinase, exon 2 is missing in this gene that you want to study. The only thing is the Cree recombinase the gene is present in all tissues, but it's under the control of a tissue or cell type specific promoter. So that means it's expressed, for example, in liver, heart, lung, or whatever, depending on the promoter. In all the other cells, tissues, it's not expressed. So because of this cell type specific promoter, Cree recombinase will be expressed, for example, in heart cells, and then you would see splice out of exon 2. So only in these cells, Cree recombinase will be produced. Cree recombinase is recognizing these LOXP sites and splicing out. In all the other cells, the gene is present but not expressed, so nothing happens in the other tissues. So you just want to memorize cell type specific promoter, that makes the recombinase expressed in specific tissues. You may, as I said, you may use this method also if you have, for example, tamoxifen inducible promoter, and you add tamoxifen to the medium, and then you would or to the food, and then you would create this splice out. So this is when, for example, a gene is involved in development and you want to induce a gene later in the life cycle then. But very often we use also this, for example, creating knockouts in heart cells in the heart using a cardiac specific promoter. So again, LOXP sites, so the target flanked gene is present in all tissues. The Cre recombinase, the gene, is present in all tissues, all genomes, but only it's expressed in a certain tissue, the Cre recombinase, because of the cell type specific promoter. So, okay. So this was Cre-Lox system. A newer system, I mean, this is an exploding field, is with the CRISPR-Cas9 system, where you can knock out individual genes, you can make mutations. It's based on the knowledge that bacteria use a system to memorize when they were infected by foreign DNA. So like a bacteriophage introduces foreign DNA into cells, and the bacteria created a system to memorize, oh, we were infected before with this bacteriophage, and to have a faster response to this exposure. The DNA gets incorporated, the foreign DNA gets incorporated into the genome, and then after exposure again to the foreign DNA, it will be faster expressed so that the foreign DNA can be inactivated much faster. So in these steps, there would be a nuclease involved that makes digestion of the foreign DNA into smaller pieces. These will be inserted into the genome. And so to say it maybe short, once it's again exposed to foreign DNA, RNA, for example, the RNA could play a role with interference with this foreign DNA, or actually also DNA. But it's a whole system is like used as a memory to be prepared faster if it's exposed again to foreign DNA, these cells. This system was now used to make it used to establish mutant organism. What you need is a nuclease and not primers, various RNA molecules that will target the nuclease to a specific site. So the application for these systems is, I mean, ultimate application would be human therapies, for example, to correct mutations in the genome. There could be, I think, like muscular dystrophy. I think they already corrected this. I mean, it's already, you can see here from the timeline, it's about maybe 10, 15 years or so, 20 years. And the crucial step in this scheme is that the nuclease that creates a cut in DNA at the site where, say, for example, the gene correction should be occur, so that this nucleus is placed at the exact site where it should act. If it's acting somewhere else in the genome, it cannot be used for human therapies. So that's clear. So, I mean, principle would be there are different RNAs that will guide the nuclease to the place of interest. So, I mean, this is like a scheme, like you have different plasmids that you will transform the cells where you want to make the mutation, and you have the RNA that will anneal to the that will introduce the mutations, guiding RNAs, and then you have the nuclease. So this complex together, nuclease with the RNA, goes to the site of interest and makes a specific cut in DNA. And that allows you to either do a modification in the DNA or introduce some other DNA, for example, some other DNA fragment. So again, the crucial step is having the cut with the nuclease, Cas9, very specific. It needs to be really site-specific. So applications, you can see here, this is 2013, for example. Usually when you create a knockout mouse, this takes months of work, just doing one knockout. In this case, you can create multiple knockouts at the same time. So it saves you, it saves you a lot of time. You can do a one-step generation of mice with multiple mutations. So lots of time saved. So this is CRISPR-Cas9 system, changes the DNA. That's what you see here on this slide here, is you use a way to interfere with the RNA, messenger RNA. And you do that with a specific messenger RNA. You can reduce the messenger RNA of a specific gene with, it's called small interfering RNA, siRNA. And so you bring double-stranded RNA into the cell that will interfere with the specific messenger RNA. It's not all messenger RNAs, it's just one specific messenger RNA, and this will be degraded. This method does not change the chromosomal DNA. It's only affecting RNA of a specific gene. So just that you know, by RNA interference means that a specific or a messenger RNA of a specific gene will be reduced in amount. The DNA itself of this gene is not affected. Okay, last one would be transgenic. Means that you can, for example, make a gene or a protein overexpressed. You can also express, for example, a mutant gene, overexpress this. That's another way. But usually it's just bringing in a piece of DNA that's expressed. And so you make there are two ways, transient transfection, or that's a plasmid that you bring in, it's stable, maintained. For transient, um, only certain cells will obtain the plasmid. Usually you can use this method when this plasmid also expresses a um, uh, green fluorescent protein. So under the microscope, you can easily identify those cells will glow green, and those will contain then also the other information that you want to express. So here you have a cDNA that's expressed, and you bring this into just a portion of the cells, not in all cells. In contrast with in contrast with a method that will maintain the plasmid in all cells, you need also a marker gene, in this case neomycin resistant gene. So in the presence of this drug G418, is an antibiotic, you force cells, or you identify sorry. Oh my God. Sorry about that. Those all cells will contain the plasmid. So this is the same principle that you use for like what I said with bacterial cells, with the with the with the with the sorry, this is had a cold. With the antibiotic resistance gene, it's the same principle, but just in mammalian cells. Yeah. So plasmid DNA, some people, they use viruses. So in this way, you can infect cells using recombinant, for example, lentiviral particle. Those lentiviral particles, or say the DNA, will be inserted into the genome. You have different components to make the virus, and you have a vector plasmid with the information what you need. You have other plasmids that are needed to make the virus, packaging plasmid, viral components. Usually you make this you make these viral particles in kidney carcinoma cell lines, 293 cells. It's a standard procedure. You let viral particle produce, and then you can take, for example, supernatant of the cell culture. You have dishes with the cell culture, and you take the supernatant with the viral particle, and then you expose this viral particle to the cells of interest. Then they will infect your cells, and then the lentiviral particle will be introduced, will infect the cells. So the lentiviral particle will insert into the genome. Another way would be adenovirus particle. They would not, for example, they would not insert in the genome. They are also not very stable in cell lines. Yeah, this uh, this is just because we say COVID vaccines, and so a new method. Uh, Well, new method. I mean, these are um, to introduce the mRNA that it's important to induce the vaccination. So this will be delivered by liposomes or lipid nanoparticles, like little balls of membranes, and they contain the cargo, the RNA. This will fuse with the cell membrane. This is called endocytosis. And the particles release the mRNA, and then they will, in this case, immune cells expose the produced protein on the surface. So this method can also be used to infect cells with mRNA, and, for example, to help, for example, with knockout of genes and so, or to reduce certain gene products. It would be another way to, okay. Once you create, say, knockout, sorry, or transgenic, you want to know, is a gene really um, low expressed, especially when you use siRNA, you want to know, is the expression level very low? You can do this. I mean, nowadays you would use RT-PCR, reverse transcriptase PCR, but some people, they have used, for example, way back, northern analysis, running out RNA on the agarose gel, transfer to an RNA membrane, hybridizing with a specific probe, for example. So this would be, but again, nowadays you would use reverse transcriptase that will produce DNA from the RNA, and then you will do a PCR reaction. If you want to locate, for example, RNA in a specific area of the tissue, you can do RNA in situ hybridization. So that's would be another way to locate RNA at specific sites, either in the tissue, but also in the cell. You remember, yeah, RNAs, for example, can be localized in the cytoplasm at, for example, at the plasma membrane, or it doesn't just float around. So you can localize it specifically in the cell. Yeah, this is also one other method to study gene expression throughout the genome, is to use microarrays. This microarrays, so you would use um, you have, for example, two different conditions. You isolate the RNA, and then you would form DNA using, again, reverse transcriptase. You remember DNA, and then you have two different labels, DNAs that you will hybridize the microarrays. Microarrays are on a glass support. You can print uh, the little DNA fragments representing all the different genes. So you have on a small area, you have all genes, for example, of the human genome. And then once you hybridize the labeled DNA coming from these two different conditions here, it will bind, it may bind only to specific areas. So that means if it's binding only to specific genes, so that means only these genes were expressed during these conditions here, fibroblasts without serum or with serum, for example, in this case. So yeah, I mean, this is how it would look like, this plate with all the genes of the genome, and you would see different colored places that would tell you if a gene is, for example, expressed or not expressed once cells are exposed to serum or without the serum. So that's, I mean, in today's world, you would not really use this method anymore. You would do RNA sequencing directly. So you can do this, whole genome RNA sequencing that replaced basically this method. Okay. So exam will be same type. You are already familiar. Okay, so 70 questions, each lecture 10 questions. So last slide, this is not, I will upload maybe this. This is just to remind you that so for a makeup exam is only possible with these specific situations here. So we were approached by people with all kinds of reasons to postpone the exams. This will not be possible. Okay, so there's only significant illness. You need to provide a doctor's note. Okay. Number two, I hope that's not the case. I mean, that was the case. One of your colleagues had this death in the immediate family. Or number three would be interviews, would be if you have this on the same day or week. So also you need evidence before. If you don't bring this evidence, main exam, okay? And presentation of a meeting, for example. Another one would be jury duty. I don't know if you need to go to court, but so yeah, because some people your priority is to finish your courses and not other things. So there are 180 colleagues. You have 180 students in this course, so we can't do. And another thing is makeup exam. So main exam is on Wednesday. Makeup exam is most likely so Friday. It will not be in the following week. So just make sure that for example, if your computer doesn't work, that wouldn't qualify for a makeup exam. You have to make sure your computer is working and you can upload your exam. So just to make sure. So I hope I'm clear about this, and good luck for your exam. Okay, thanks. Okay, sure. Thank you. No, it's take care. Okay. Good night. Take care. Good luck. Good luck. Listen.